So the problem of getting genetic material into cells has been around for a while. There are a few ways of doing it. The clinically approved methods, so now we're talking about these engineered cell therapies. What's done there is they actually engineer a virus. So you engineer a virus. The great thing about a virus, viruses have evolved over millions of years to be able to infect our cells and put DNA into our cells. And so scientists for decades now have been able to engineer viruses to get those viral cells to put in DNA that we want. And so one way of doing this engineering is using viruses. Another way is what you might call non-viral techniques. And these non-viral techniques require two parts. You need some physical mechanism of opening the cell, and then you need some molecular biology in order to do the actual gene editing. And that's where things like CRISPR come in. So with the advent of CRISPR and the ability to edit the genome, either knock things out or put things in, coupled with physical mechanisms for opening the cell, you can now have an alternative to viral. So previously all you could do was viral, but now there are gene editing methodologies like CRISPR. CRISPR isn't the only one, but it is the most popular and one of the easiest editing tools out there. Coupled with non-viral delivery techniques, you can now compete with viruses. And there's a big advantage to this. So in this field that I mentioned where there are already some approvals for engineering cells for cancer, there's a 2-year wait period in order to produce viral vectors. And so if you today devise a new therapy that leverages these viral vectors, it's 2 years before you can actually get that vector manufactured. There's not enough capacity in the industry. In addition, it's very expensive. So these therapies are several $100,000, and the cost of goods sold, about half of it is in just engineering the virus. And so if you can eliminate this virus or remove the need for the virus, you can cut down the cost, you can cut down the time for development, and hopefully get these therapies to patients faster. But there are some benefits of viruses. Viruses, they work. They work reasonably well. The approved gene therapies that you've heard out there, all of them use viruses today. And so there's still a technological hurdle to prove that you can couple these non-viral editing techniques like CRISPR with non-viral delivery techniques like what my lab works in order to compete with viruses. But if you can do it, there's a lot of cost savings potentially in the industry and benefits potentially for patients as well. I think the biggest problem in the area with viral is the amount of time it takes to scale up, and it's also unpredictable. If you think about it, to engineer a virus, this is a whole bioengineering platform that you have to engineer and scale up. So what's done at the bench doesn't scale to the lab very well, and that's the big problem. At small scale, you can do things that are very challenging to do at large scale for healthcare. And so there's a roughly 18-month time period to scale something that you could do on the bench all the way up to GMP manufacturing for a therapeutic. And it's very difficult to overcome this lack of scalability. And an advantage of the technique that my group has been working on is that the physics scale very well. They actually scale with time. And so when we're doing something at the bench, the time it takes for our editing is on the order of fractions of a second. And when we need to do clinical scale, we can generate the therapeutic payload for someone my size in less than a minute. And so you just wait a little bit longer and you get enough. Whereas a technique like making virus doesn't scale in the same way.